I slightly changed my title because I realized that I wouldn't be able to talk about everything I want to talk about in the short time. So do I have, like, how much time do I have? Oh, Tilo, you're muted. Sorry.
20 minutes plus
Thank you.
A little bit time for questions.
Perfect. Okay, so today I would like to talk about a recent work that we did with Merav Stern, that many of you know, and Nico Strate, who's the first grad student to defend from my lab. And this is about how a big heterogeneity of timescales can arise from chaotic dynamics in random neural networks, and this reveals like a new kind of metastable phase of chaotic dynamics that we're very excited about. And so before I start, I first want to highlight the people who did the work in my lab. And so this is the lab, and the people who did the work are Merav Stern, who's a Hebrew now, and Niku, and this is our preprint that's now under revision, and I encourage everybody to look at it if you want to know more about it, about the details. So the kind of work that we do in the lab is about studying temporal dynamics and temporal organization of behavior and neural activity, and so we look at this relationship between behavior and neural activity in both kind of incoming sensory input that drives activity in sensory cortex, and also outgoing motor activity that generates behavior. And we're interested in this kind of the temporal organization that can be kind of approximately organized in terms of three different axes of temporal variability. The main thing we want to talk about today is the hierarchical structure, such that many timescales are present in behavior at all times. So from 100 milliseconds to seconds to tens of seconds to minutes to hours and years, even for humans. Although we study other directions of variability, which is contextual modulations of these timescales, and how at every level of this hierarchy, temporal hierarchy, there's like an inherent stochasticity in behavior that arises, we believe, from metastable dynamics. And so we study the neural mechanisms of these different these different temporal variability axes. And in particular, today we're going to focus on how a hierarchy of timescales might arise from some simple recurrent neural circuits. And in particular, we will see that we will propose that in order to generate neural activity that simultaneously vary on many different timescales, orders of magnitude apart, the simplest mechanism we can think of in terms of emergent collective activity is the activity generated by neural clusters of different sizes. So this is going to be the punchline of the talk. And so why are we interested in this? Well, there's this kind of series of observations from experimental studies that point to large heterogeneity of timescales across cortex. So if we think about the autocovariance function, the spike autocovariance of single neurons, this is what I showed you here on the left. You can estimate the timescale, the typical timescale in many ways, as the full width at half max, or in case of this, in the case of this paper on the right from the Xiaojun Wang's lab, in terms of the average decay time of population activity. And what they found in the monkey cortex, awake monkey, is that the average timescale of population activity, multi-unit activity varies along the cortical hierarchy from very fast timescales in the kind of visual stream and the occipital cortex to kind of much lower timescales as you move towards the frontal cortex. And they vary from 50 milliseconds to 350 milliseconds. So this is the average timescale of autocorrelation within different areas. What's even more striking is the fact that if you focus into one single area, now, for example, the anterior cingulate cortex, and this is work from Johnny Wally's lab, what they found is that different neurons within the same circuit have very, very different time scales, again measured from their autocorrelation during spontaneous activity, that vary from, let's say, 10 milliseconds all the way to 1 second, which is the longest time scale they were able to measure, to estimate in their data set. But in our hands, it extends even to longer time scales. So this huge heterogeneity of time scale is seems to be a crucial feature of brain activity that reflects also the structure in naturalistic behavior. And so our question is, so what's the simplest mechanism that could give rise to this heterogeneity of timescales in terms of recurrent dynamics? And so kind of a fun, a simple model to study this from first principles, if you want, is a network of recurrently connected rate neurons, rate units. And so that's what we started from. And so our point of view here is a phenomenological approach in which we think about every unit in this recurrent network has a population of neurons. So we think about this as kind of a phenomenological model of some cortical activity. Every unit represents like the average activity in a mean field sense of a bunch of neurons that have the same functional properties, for example, some kind of a Hebbian assembly. And so I wrote here on the bottom left the usual equation for the dynamics of these networks. And so UI is the activity of one of these neural clusters. And they're all coupled together with recurrent couplings, and they're usually taken from Gaussian distribution with mean zero and variance G squared over n. This is the usual parameterization of a random neural network. And the extra ingredient we're gonna focus on is a self-coupling term that you see here that we call S, which in our network is of order 1. As opposed to the usual kind of Crisanti formulation, where this is order 1 or square root of n. And so S represents, roughly speaking, the product of the size of this neural cluster times its own recurrent coupling, if you want. And so this is a model that was proposed by Merav several years ago, and it's kind of super interesting in its own right. But what we're interested in today is the autocorrelation, to study the autocorrelation function of this model. And so this model has like an interesting dynamical picture with different phases. And there's one phase in which this model displays chaotic activity that I showed here on the right. So these are 3 example units from this model. And so if we take the connectivity such as here, in which the off-diagonal parts, again, are these JIJs that are taken randomly from this Gaussian distribution. And we can fix the self-couplings. So the self-couplings are the diagonal in this coupling matrix here, in this connectivity matrix. We can start from a simple example in which all the self-couplings have the same value. Here, for example, is like 3. And what we find is that we can measure the autocorrelation of all the neurons in the network, of all the units, in the network. The units represent these neural clusters. And what we find is all the units have the same autocorrelation. There's no heterogeneity of timescales here in this network when all of the self-couplings are equal. And so this is the autocorrelation of the activity. And we see that the histogram of coarse neurons is very, very tight. So there's no heterogeneity. Okay, so this doesn't, so the simple network like this doesn't work, so doesn't have any heterogeneity of timescales, which is what we were at, what we're trying to model. And so, well, you guess where I'm going. So now what we take instead is we take now different self-couplings for different subsets of the units. Let's say half of the units have one particular self-coupling. The other half of the unit has a different value of the self-coupling. For example, this top left half have they have self-coupling equals 1, and the bottom right part has self-coupling equals 4. And now we can calculate the autocorrelation function of all the units with one self-couplings and all the units with the other self-coupling. And what we found is that, lo and behold, this works. So a network with where units have different self-couplings produces different timescales. And so in particular, the timescale of one unit depends on the self-coupling, on its own self-coupling. So in this case, we have two groups, one with small self-couplings that has a small timescale. We define the timescale as the width a half max of the autocorrelation function, if you want. And the other population with a large self-coupling has a large timescale. So this kind of works. So this idea then that neural clusters of different size generate different timescales simultaneously within the same network. And we can understand all of this, all of these dynamics in this network in terms of dynamic mean field theory, if you want, where we replace here the input to a particular unit coming from the recurrent couplings with all the other units in the network with a Gaussian mean field, and we can write the self-consistent theory for this. And what we find is that, well, so this theory has a lot of other interesting features. For example, as a function of the two self-couplings in the two groups here of neurons, we have a complicated phase diagram where in some regions we have chaos, and in some regions we have transient activity with fixed points. I just wanted to give you like an example of what this looks like. So if the self-couplings are, one self-coupling is small and the other is large, here in the region 1, we have transient activity with kind of the small self-coupling population fluctuates around 0. The large self-coupling populations fluctuates around large values. And yeah, there's a very cool phase diagram, but I'll skip it for now. And so what's interesting is that we can focus on these chaotic activity regions, because that's what we want to look at. We're interested in this autocorrelation function in the chaotic regime. And if we fix one self-coupling to be S1 equals 1, if you want, this one, and if we increase the value of the self-coupling in the second group, then we can calculate the ratios between tau2 and tau1, the self-couplings in the two populations, and we see that this ratio increases exponentially as a function of this ratio of the self-coupling. So there's a parametric separation of scales in this network that we can tune by changing the ratio of the self-couplings. So, okay, so this generates two self-couplings, but we want to have some network that generates the full heterogeneity of timescales that was observed in the data by Kavanaugh from the Wallis lab. And 4
minutes roughly.
Oh no, 4 minutes only. Okay,
okay,
so
5.
okay, cool. I go faster. Okay, so how do we do that? Well, we take a distribution of self-couplings. For example, we take a log normal distribution of self-couplings here on the top, and what we find is that we find the long tail distribution of the log of the timescales, right? So which kind of matches this heterogeneous distribution of autocorrelations function from the Wallis lab. So okay, so where's metastable activity in this game? So what we found is actually something very interesting, that when the self-coupling becomes very large, Then the network activity, so what I'm showing you here is a snippet of network activity for a unit that has a very large self-coupling, equals 5, for example. Then the network activity becomes metastable. So the activity is localized around 2 values, plus or minus the value of the self-coupling. And it has these very long-lived bouts where it's localized and it fluctuates around those values. And then it abruptly switches to the other value, and so on and so forth, such that the distribution of dwell times around these plus and minus S2 values is very long tail. And so we can study this particular regime, again, using mean field theory in the probe approximation when we take one neuron with very large self-coupling and all the other neurons with the same self-coupling S1 equals 1 if you want. So we have an effective theory for this probe neuron, which is described by this kind of Langevin equation, where the noise is the Gaussian mean field that has a particular autocorrelation function, which is the average autocorrelation function of all the other neurons in the network. So it's kind of a Langevin equation driven by color noise. And okay, you might think, okay, we have a Langevin equation. Let's study the Fokker-Planck description for this to capture the distribution of values and also the transition times. And it turns out that if you use the naive Fokker-Planck equation that you will get from this, but with white noise, where this was not the actual self-consistent mean field, but this was just the white noise, then this doesn't work. So what I drew here is the histogram of the values of this of the activity of this probe neuron in blue, and overlaid with the solution to the white noise Fokker-Planck equation, if you want. And you see that it's completely wrong. It doesn't work. So in dashed line, you see the potential, which is the integral of this blue box. And with the red, you see it doesn't work. So it turns out that there is what's called a universal color noise approximation to the Fokker-Planck equation, which is an extension of Fokker-Planck that that allows to consider color noise, so noise with a non-trivial autocorrelation function. And you can actually use this universal color noise approximation to calculate a correction to the effective potential in the Fokker-Planck. And what we found is that that works very, very well. And the effect of the color noise, which is the mean field of all the other neurons in the network, is that it shrinks the distribution of activities that you will get with the white noise. And so this is the full exact description with the universal color noise approximation that we are very excited about, because it's very different from the white noise. And we propose it as kind of a new interesting way to think about recurrent network dynamics, if you want, that captures this metastable phase. And from this Fokker-Planck, we can calculate the mean first passage time between these quasi-stable states in this metastable regime. And it turns out that we can calculate it analytically, and we find that the logarithm of the average first passage time goes with the square of the self-coupling. So this, the time scale of these neurons is exponential in the square of the self-coupling. And for large self-coupling, it matches very well the simulations here, and the universal color noise approximation kind of goes well. And so okay, 1 second. Okay, 2 minutes. I'll finish this. We show that this captures the dynamics of now spiky networks of excitatory inhibitory neurons arranged in cluster, where if you do the mean field, you find that a self-coupling arises from the recurrent interaction of neurons within the same cluster. And if you now simulate network of EI clusters with a hierarchy of cluster sizes, you find again here on the y-axis that the log of the average dwell time is quadratic in the self-coupling. So this matches the qualitatively the results from recurrent network dynamics. Okay, so something else. Okay, I don't have time to show you, but it's in the paper. What is this good for? Well, we can use this network with multiple timescales to do a demixing of broadband inputs using this idea that Kanaka-Rajan had of suppressing chaos. But yeah, I guess I don't have time to show you that. I can show you if you ask me a question. So let me wrap this up. So again, this is work by Merav and Niku. So we have a distribution of self-couplings that generates chaos with a heterogeneous distribution of timescales across clusters. In this particular limit of large self-couplings, we recover a metastable regime where we can study with this correction to the Fokker-Planck theory. And this reproduces this kind of metastable dynamics that we studied in the lab for many years, which is this kind of cluster etc. inhibitory spiky networks. And this is a cool application in terms of the mixing broadband inputs. And yeah, that's it. I can stop here. I'm super over time. Sorry about that.
No, that's okay. You just started later, but there's a plenary talk also coming next. That's why I was a bit rushing. Yeah, thank you very much for this